Howdy, my name is Lauren, and over the past few weeks, I have done research about detrimental chemicals in pesticides, one of them being imidacloprid, and how it can cause accelerated senescence, cell damage, and premature cognitive dysfunction in Apis mellifera. To begin with, Apis mellifera is a species of bee that is native to Europe, Africa, and the Middle East, and has been introduced to every continent, save for Antarctica, and is mainly referred to as the western honeybee in the United States. Imidacloprid is a systemic nicotinoid insecticide that controls pests by mimicking nicotine to disrupt their central nervous system, often leading to paralysis and eventually death. However, before there is death, there is something else. While there are innumerable chemicals that can be used to create problems for bees, imidacloprid is one of the worst. Through research, it has been found that imidacloprid was most effective in restricting the movement of bees when they were exposed to it via contact and/or consumption. Having come into contact with this chemical, bees experience something called oxidative stress. This is when there is an imbalance between the reactive oxygen species, or ROS, and antioxidants. And since the antioxidant system plays a crucial role in protecting the brain and neural tissues from damage, this lack of protection leads to premature cognitive dysfunction. The lack of cognitive function means more restricted movement in the muscles of bees, such as their thoracic flight muscles, which are part of their main mode of transportation, flight. In this figure, there are two main groups, 3 groups of feral western honeybees and 3 groups of managed western honeybees. Each group was suffering from oxidative stress from contact with imidacloprid, and you can see that the feral groups were much better off with handling that kind of stress, which makes sense. The managed bees are much more likely to be closer to managed crops, those of which would have some sort of insecticide or pesticide to ensure no loss of crops due to insects. Unfortunately, bees are insects. If the flowers were open at the time of spraying the pesticide, the bees would come into contact with it much more often, rather than the feral bees who are more exposed to farther away natural flowers with no kind of chemical on them, organic or otherwise. It is even worse when the agent imidacloprid is combined with other detrimental chemicals. In this figure, Apis mellifera is not only exposed to imidacloprid, but also glyphosate and difenoconazole. Then, in addition to single-agent exposure, these chemicals were combined with each other, imidacloprid and glyphosate by itself, and so on and so forth. This figure shows the mortality rate of western honeybees after 16 days after the initial exposure to varied chemical amounts. The effect of these chemicals can not only increase the speed of cognitive dysfunction, but also functional senescence. Senescence is when a cell permanently stops dividing and enters a dormant stage without dying. This in itself is a natural thing and usually happens to stop the growth of tumors. However, contact, or in this case consumption, this process is sped up significantly and sometimes noticed around the very young age of 18 to 28 days. This next figure shows the level of protein damage through carbonyl content assay at day 9 after Apis mellifera groups were exposed to various treatments, one of them, as you can see, being paraquat dichloride, otherwise simply known as paraquat. Oxidative stress can also cause stress in other systems of a bee, like the endoplasmic reticulum, which, if under enough stress, can trigger the literal unfolding of proteins and eventually induce a type of cell suicide called apoptosis. High ROS levels can also cause damage to the mitochondria and cellular membranes, causing damage at the cellular level. Now, how does imidacloprid-induced oxidative stress influence the biology of Apis mellifera? In all, this oxidative stress plays a critical role in altering western honeybees' biological function, for the worse at both the cellular and single organism levels. The resulting imbalance between ROS and antioxidant defenses contributes to progressive cellular damage, particularly with neural and metabolic systems. These physiological disruptions make themselves known through a decline in cognitive function, reduced motor performance, and accelerated functional senescence. Collectively, evidence suggests that oxidative stress is a key mechanistic pathway through which imidacloprid and other detrimental compounds found in pesticides shows its toxic effects on Apis mellifera health and longevity. The significance of this research lies in its potential contribution to better understanding the biological pathways through which neonicotinoid exposure, specifically imidacloprid-induced oxidative stress, affects the western honeybee. By linking molecular level disruptions such as reactive oxygen species imbalance to broader physiological outcomes like cognitive decline and increased and quicker mortality rates. These findings are particularly important given how bees are just about one of the most important insects in the world as global pollinators and ecosystem stabilizers. Ultimately, this research highlights the need for more comprehensive pesticide assessments that not only account for the lethality of it, but also for the long-term biological and ecological consequences.